Namaste children. Today we will start a new chapter, that is chapter number 12, Biotechnology and its applications. In the previous chapter we discussed about the basic principles and processes of biotechnology, that how rDNA that is produced and how this rDNA it is transferred into the host cells through the transformation methods, and how the bioreactor it works for the production of the desired products in the large quantities. Okay, so let's discuss in detail. So first we will discuss about that what are the three critical research areas for biotechnology to work. So first, that is providing the best catalyst. Providing the best catalyst means that like in a bioreactor, a catalyst or an enzyme that is used like the microorganism for the production of the desired product, because these microorganisms they are responsible for converting the raw materials into the desired product. Now next, that are creating optimal conditions. Creating optimal conditions means that optimum pH, optimum temperature and other optimum conditions that should be provided. Okay, so that the microorganisms or the other plant cells or animal cells which we are using as a catalyst, so they will be able to grow properly in that medium. Next is downstream processing technologies. Downstream processing technologies means that after the production of the desired product, so how this product that is separated, isolated and then purified and then released in the market for the commercial purpose, commercial sale purpose. So all these steps we have discussed in the previous chapters also. So in this chapter we will discuss about the applications. So first we will talk about the plants, that how the biotechnology it is useful in case of plants. So generally, if suppose we will talk regarding the increase of the food production, so how the food production that can be increased or that is increased in the past days. Okay, so first point, that is the agro based agriculture, second that is organic farming, and third that are genetically modified crops. Okay, so basically these three methods are there which are responsible for increasing the food production, for increasing the crop yield. Okay, so it is also responsible for the green revolution. Clear? So now first point, that is agro-based agriculture. Now what is agro-based agriculture? Agro-based agriculture means that the use of the fertilizers, use of herbicides, pesticides, means use of the chemicals for increasing the food yield. Clear? But there are some disadvantages also of this method. What are the disadvantages? Yes, because they are harmful for the environment when they are used in the excessive amount. Clear? And second, that is the organic farming. Organic farming means that using the microorganisms as biofertilizers. Okay, in place of the chemicals, if we are using the microorganisms as a biofertilizers, so that is known as organic farming. And third, that is GM crops, means genetically modified crops. They are also very helpful in increasing the food production. Okay, so out of these, if we will compare, then genetically modified crops, that are the best methods. Okay, these are the best method to increase the crop yield. Now next we will discuss about the applications of genetically modified crops in agriculture. But first we will talk about genetically modified crops, that what are actually GM crops. GM crop means the transgenic plants in which the gene, means the foreign gene that is inserted. Okay, and that gene that is inserted to get the desired character. So that plant is known as genetically modified crops. So let's discuss about the applications of these kind of crops. So first, that is more tolerant to abiotic stresses. Means we can produce such kind of plants which show more tolerance against the abiotic stress. Abiotic stress means that if suppose the external condition the external environment having more heat, cold, drought, etc. Okay. And second, that is pest resistant crops. Pest resistant crops means that we can produce such kind of crops which show resistance against the pest, and then that pest will not be able to attack that particular plant. Next, that are insect resistant crops. So similar, the insects, they are not able to attack that particular resistant variety of crops. Next, that are the increased mineral level. Increased mineral level means that the mineral efficiency of the plant, it will be increased. Okay, so it prevents the early exhaustion of the fertility of the soil, which occurs usually. Now next is enhanced nutritional value of food. Means the vitamin content of the plant, it can also be improved through this method. Now children, next we will discuss about the BT cotton plant. BT cotton plant, it is an example of genetically modified crop, and how the cotton plant that is genetically modified by the insertion of the BT gene, that we will discuss today. Okay, so let's see here that this bacteria, it is known as Bacillus thuringiensis, that is BT. Clear? And this contain a BT gene in it. It contain a BT gene in it. So when this BT gene, it is isolated and then it is cloned in a vector. Clear? Till here it is clear to all of you that when it is cloned in the vector, that it is known as rDNA, that is recombinant DNA. And when this recombinant DNA, it is transformed into the plant, then this plant, it will become BT cotton plant, which shows resistance against the insects. Okay, certain insects. Why? Because this gene, it is responsible for producing the insecticidal protein, which is toxic in nature and which kill the insects. So let's discuss about this gene. This BT toxin gene, it is also known as cry gene. And cry gene, it is also of three kinds. That is cry 1AC, cry second AB, cry 1AB. Okay. So these genes, like first AC and second AB, they show their response against the bollworms. Okay, they control the bollworms attack. And cry 1AB, they controls the corn borer. Clear? Even these cry genes, they are responsible for killing so many insects also, like lepidopterans, coleopterans and dipterans. Lepidopterans, that are like tobacco, birdworm, armyworm, coleopterans, beetles, dipterans, flies and mosquitoes. How does this happen? How the BT gene, it is responsible for killing certain insects and make the plant resistant against these insects. So let's discuss in detail about the mechanism of this BT gene, that how does it work. So now we will discuss the mechanism of BT toxin gene, that how it is responsible to kill the insects. Clear? So let's discuss in detail. BT toxin gene, it is inserted in the plants in the form of rDNA. But why this gene, it is not harmful in case of bacillus and in case of plants? Because when after transcription and then after translation, when this gene, it produces the toxic protein, then this protein, it is harmless. Why? Because it is present in the form of protoxin, means inactive. Okay, but when the insects, they engulf this protein, but when the insects, they engulf this protein, then what happens? This protein, it will become active in the alkaline pH of the gut region of the insects. This will become active in the alkaline pH of the gut region. Clear? So when it will become active, then what happens? It binds with the midgut epithelial lining. Okay, and then it creates pores. So when pores that are created in the epithelial lining because of this toxic protein, then what happens? Swelling will be there. Cell swelling will be there, and then cell get cell lysis, and then ultimately it cause death of the insects. It cause death of the insects. So in this manner, this Bt toxin gene, it produces the toxic protein which is harmful for the insects or which can kill the insects.